Disease-specific vaccine design, tuberculosis. Vaccine design means the scientific process of creating a vaccine that can safely and effectively trigger immune protection against a particular pathogen, virus, bacterium, or parasite. Vaccine design is the systematic approach to identify antigenic components of a pathogen and formulate them into a vaccine that induces protective immunity without causing disease. Goals: to generate long-lasting immunity, memory T and B cells. To neutralize or eliminate the pathogen. To minimize side effects and enhance safety. Disease-specific vaccine design. Disease-specific vaccine design refers to customizing vaccine components based on the pathogen's biology, structure, life cycle, virulence factors, the immune mechanisms required for protection, humoral or cell-mediated, host pathogen interactions unique to that disease. For example, influenza vaccines target variable surface proteins, HA and NA. HPV vaccines target viral capsid proteins, L1. Tuberculosis vaccines must elicit cell-mediated immunity, not just antibodies. Designing a vaccine against tuberculosis, TB. Background: causative agent, Mycobacterium tuberculosis, MTB. Current vaccine, BCG, Bacillus calmette-guerin, a live attenuated strain of M. bovis. Limitation: effective in children, prevents severe TB forms, but poor protection in adults against pulmonary TB. Hence, modern vaccine design aims to create more effective disease-specific vaccines for M. tuberculosis. Pathogenesis of tuberculosis. Understanding TB pathogenesis is critical for vaccine design. Stepwise pathogenesis. Entry: MTB enters via inhalation of aerosol droplets containing bacilli. Infection of macrophages. Alveolar macrophages engulf MTB via phagocytosis. But MTB inhibits phagosome-lysosome fusion and survives inside macrophages. It resists killing by preventing acidification and producing antioxidant enzymes. Immune response. Infected macrophages release IL-12, TNF-alpha, IFN-gamma, recruiting more immune cells. Granuloma formation occurs, a hallmark of TB. Central necrosis, caseation. Surrounding macrophages, lymphocytes, and fibroblasts. MTB persists in a latent state inside granulomas. Reactivation. When immunity wanes, bacilli reactivate and cause active TB disease. Immune response against TB. Protective immunity is mainly cell-mediated. Key cells. CD4-Th1 cells, secrete IFN-gamma, activate macrophages. CD8 T cells, kill infected macrophages. Antibodies have limited role. Therefore, vaccines must induce strong Th1 type immunity. Steps in designing a vaccine against TB. Step 1. Identify protective antigens. Select proteins that induce strong T cell responses. ESAT6, early secreted antigenic target 6. CFP10, culture filtrate protein 10. AG85 complex, AG85A, AG85B. PPE family proteins. Heat shock proteins, HspX. These are expressed during infection and known to elicit IFN-gamma mediated immunity. Step 2. Choose vaccine platform/type. Different vaccine platforms are being tested against TB. Vaccine type. Example. Features. Live attenuated. BCG, existing. VAC, new, attenuated MTB. Mimics infection, strong CMI, but safety concerns. Subunit. H56, M72/S01E. Safer, well-defined antigens, require adjuvant. Viral vector. MVA85A, modified vaccinia virus expressing AG85A. Strong Th1 response, good boosting candidate. DNA vaccine. DNA encoding AG85A, ESAT6. Easy to design, induces both humoral and CMI. mRNA vaccine. Experimental. Similar to COVID-19 vaccine approach, encoding TB antigens. Peptide-based. Synthetic antigenic epitopes. Stable, focused immune targeting, low immunogenicity. Step 3. Select adjuvants and delivery system. To enhance T cell responses. Adjuvants, AS01E, used in M72, MPLA, CpGODN, CAF01. Delivery systems, liposomes, nanoparticles, viral vectors, microneedles. Step 4. Preclinical evaluation. Test in animal models, mice, guinea pigs, non-human primates. Evaluate IFN-gamma production, granuloma formation, bacterial load. Step 5. Clinical trials. Phase I, safety. Phase 2, immunogenicity. Phase 3, efficacy against TB in humans. Example, M72/S01E, by GSK, showed 50% protection in phase IIB clinical trials, a major success. Example, modern TB vaccine candidate, M72/S01E. Feature. Description. Type. Recombinant subunit vaccine. Antigens. MTB32A plus MTB39A fusion protein. Adjuvant. S01E, liposome based with MPLA and QS21. Immune response. Strong CD4 T cell and IFN gamma response. Efficacy. 50% protection in adults with latent TB infection. Status. Preparing for phase 3 trials. Rationale for disease-specific TB vaccine design. Because TB immunity depends mainly on Th1 cell activation and macrophage killing, the vaccine must deliver antigens to MHC class I and II pathways, sustain long-term memory T cells, possibly target latent antigens, example HspX, for preventing reactivation. Modern tools for TB vaccine design. Tool. Purpose. Reverse vaccinology. Identifies antigen genes via genome screening. Immunoinformatics. Predicts T cell epitopes computationally. Structural vaccinology. Designs stable, immunogenic antigen structures. Systems biology. Analyzes immune network after vaccination. Nanotechnology. Develops new adjuvants and delivery vehicles. Future directions. mRNA-based TB vaccines, similar to COVID mRNA tech. Heterologous prime, boost regimens, BCG prime plus subunit boost. Latent TB vaccines, targeting dormant bacilli. Mucosal TB vaccines, intranasal or aerosolized delivery.